To the last seminar in our seminar series. Today we are delighted to have Dr. Chad Steele as our presenter. He joined Tulane in 2018 as a professor and the chair of the Department of Microbiology and Immunology in the School of Medicine. Before coming to Tulane, he was the Assistant Dean of Research Administration and a professor in the School of Medicine at the University of Alabama, Birmingham. His research is focused on understanding lung immune responses to an opportunistic fungal pathogen. He holds a BS in chemistry from the University of Louisiana in Monroe and an MS and PhD in microbiology and immunology from LSU. Dr. Steele has current and past NIH grant funding, and he has more than 100 peer-reviewed publications. He has served on many NIH study sections and is currently a standing member of the Immunity and Host Defense Study Section. Dr. Steele's fun fact is that before going to graduate school, he worked as a quality control chemist for a company that makes motor oil, antifreeze, and brake fluid. Dr. Steele is going to speak with us today about the NIH's expectations for enhancing reproducibility through rigor and transparency. Please join me in welcoming him.
Welcome, everybody. I know this is a very robust course over the last several months, and it's for some reason I just came across the lectures about 30 minutes ago, and so it turns out I've actually got some duplication slides, not some graphs and stuff like that in my presentation. So hopefully I won't bore you too much on that. But thanks to Brian and Lisa for inviting me to do this. I actually, putting this thing together over the last couple of days, I had a lot more fun than I anticipated based on this topic. But I kind of want to go over, this new enhanced reproducibility expectation or policy change. You want to come on? Yeah. Come on.
Thank you.
Yeah. So just a quick outline. A little bit of background about myself. It's always about me. That's what my wife says. So. And then, the definition of what we're talking about today. How these new NIH expectations, or you can call them policy changes, how they came to be, really the background of what precipitated them, what they are, and what I have is concrete examples of how they can be applied. So I kind of personalize this talk. If you ever seen me give a talk before and everything, I'm not really a textbook guy and, do it this way. I kind of like to show you how I apply things. And when I give lectures about, the lung and things like that, I show data from my own lab and things like that. I'm actually going to show you some data today, just to kind of, reinforce what these policies and procedures mean to a PI or an investigator. And then finally, how to apply these general concepts of rigor and transparency in your own research. So let's start off with my fun fact. So I'm a biomedical researcher. I work on mice. I work in the lung. I do fungal infections. I do fungal asthma. But my first job at undergrad was this quality control chemist in a little town in northeast Louisiana called Tulane. You can see the little star right here. It's right on the close to the about 10 miles from the border of Mississippi. Here we are down here, so about 4 hours away roughly. And I did. The lab is buried somewhere in here, if I remember correctly. This is an area that we filled all the motor oil cans and brake fluid cans and things like that. And so I did this. And the reason why I wanted to bring this up is that I think it's very appropriate for the topic of today. And the reason why is that because of this experience, you see, I only did this for a little over a year because actually in May of 1996, I moved here to New Orleans to start grad school at LSU. So I did this for a while, really liked it. I really, really loved it. I almost went to Chicago to work for AkzoNobel, which is like the 5th largest chemical company in the world. Came really close to doing that because we were working with them at this company. The second job I was looking at was actually being a chemist at a winery out in San Francisco, and I really wanted that job bad. I didn't get it all. I was talking to some other dude who had wine experience. I only had motor oil experience. So anyway, so I brought this up because through this period, to this day, this is what, 24 years ago? To this day, there's only one motor oil that I use in my car, my lawnmower, my chainsaw, whatever. And the reason why is because of water. And so what it is Castrol. So we make 10 or 15 different types of motor oil at this company. This is the only motor oil that I used to know myself. And the reason why is because this company made us do so much extra work than all the other companies. In particular, one thing in particular is this shear stability index. This is this weird, crazy machine where we would make their motor oil, we put it through here, and it would simulate going through a car for 3,000, 4,000 miles in about 2 hours. And then we'd have to do all these tests on it after. And then we'd have to print out our results on something called a C of A, or certificate of analysis. So we load this on an 18-wheeler. This 18-wheeler would actually drive this down to Baton Rouge. They'd get our certificate of analysis. They'd take a sample. They'd go do the exact same test. If it didn't match my numbers within a certain error, they'd send the truck back to us. So this company was very, diligent on how they did stuff. And so what we're talking about today, Rigor. So this is why I use these products, is because they go to the nth degree to make sure their product is optimized and the best that's out there, because they make it. It wasn't just this test. There was 3 or 4 other tests. But I think it's an appropriate story and my fun fact to illustrate how Rigor can be your friend, if you will. And I'm going to show you in a minute here, and I seem to think so too. So what do I know about today's topic? So Lisa talked about being an extensive grant reviewer. I am. One thing in my career is that I never say no to NIH. Call it politics, call it favoritism, I don't care. I just never say no to NIH. And so I have a lot of extensive grant experience in reviewing. And I put this up here not to brag. It's just because I think when you have people that give talks like this, you need to know that they know what they're talking about, or at least have confidence that they've been through it before. A lot of times you'll have somebody that gives you a grant writing workshop and they hadn't written a grant in 15 years. So, so this is applicable, and it goes on. This is all the way through next year, some stuff I have going on next year. So I've reviewed for NIH extensively. The other thing is that I've written to NIH extensively. These are only my successes. I probably have triple the number of not successes. But so I've had to not only experience this from a reviewer standpoint and know how these grants are put together, but I've had to do them myself as a grantee or as somebody who's applying for grants. And so this is just a couple of things that I've had to deal with as I've gone through all these times. The R01 went from a long to short, paper to electronic. First R01 ever written, FedEx box, 5 hard copies, mailed off the day before the grant was due. And anybody who's 15, 20 years in, that's the way it was done back then. And you got your score not by comments. They didn't have comments when I started out. You got to sit to you in a letter, and you hold up to the light and you can see your score. So it was kind of an interesting transition. Scoring system 100 to 500, which was actually really nice because you can get a 120 or a 275 or a 390. Now you can't do that. It's 1 to 9. It's not 1.5. It's not 2.5. It's 1 through 9. That's it. Critiques have changed. Strikes. This is submissions essentially. Used to we could always do 3. Now there is no such thing as 3. You only get 2 now. And then content sections. So the background is now the significance. Experimental design is now the approach. Innovation. Yeah.
For the novice, and only 3 what?
Only 3 submissions of an individual grant. So you submit it once. That's the A0.
Right.
You can revise it and submit it again. That's the A1.
Okay.
And then you can revise it again. That's the A2. We don't have the A2 anymore. They did away with it 6 or 8 years ago, something like that. So now you can only submit a grant twice. Afterwards, you got to start all over again. So you can't come back in. You can still take the same grant. There was actually a period of time where you do A0, A1, and then you had to just trash it. Completely new grant, start all over. Now they're actually letting you take that A1 that didn't get funded and repackage it with some of the similar ideas and similar aims and come back in. So, yeah. And so, and then the topic of today, the scientific premise is now rigor of prior research. This is sex as a biological variable, which I'm going to touch on it, but not going to talk about a lot because Tonette's going to talk about that next week, and then identification of reagents. So what are we talking about today? What is enhancing reproducibility through rigor and transparency? So this came out about a little over 4 years ago. So the definition is rigor is the strict application of the scientific method to ensure unbiased, well-controlled experimental design, methodology, analysis, interpretation, and reporting of results. And so the goal of this, when it first rolled out, and I'm going to show you a ton of background on why this came to be. And like I said, some of it you've already kind of heard, is that essentially the NIH is trying to promote the highest level of integrity, right? So you want to ensure that NIH is funding the best and most rigorous grants. Highlight the need for applicants to describe in detail, in high-level detail, the way they arrived at conclusions and data that previously had not been, really annotated as well as it probably could have been, and then highlight the need for reviewers to consider these details and how they review. And you see this. This is not something that is just a policy on the NIH website. This is in practice. I do study section 4 to 5 times a year, or on the regular study section 2 or 3 more, ad hoc. And we talk about these things. This is not something that we just, say, okay, NIH is making it more difficult on us again. We actually, we subscribe to this ideology. So how did this come about? So this is some perspectives that were written 12, 14, you heard about this guy previously. This is Arturo Casadevall. He's actually a good friend of mine. I've known him for 20 years because we do similar research. And I think Bruce Bunnell showed you a couple of weeks ago. So Arturo has been tracking retractions for quite a few years now. He's got 8 or 10 publications along with Ferric Fane, looking at retraction index and what are some of the characteristics of those and things like that. And so they wrote this paper in 2012. And then, like I said, I call it a call to arms. You had all these commentaries and opinion pieces from the journals, and people were, to put it, in today's climate, people were fed up. They're fed up with people who retract the stuff. They're fed up with not being able to reproduce, somebody's results and things like that. And so it kind of really, this groundswell, and NIH is noticing and, things like that. And so Bruce showed this image here. You can see that these are increasing over time here. This is only for 2011, as this paper was in 2012. But you can see the major one of these is fraud, suspected fraud for the retraction, error. I've had colleagues that have retracted papers before because they made a calculation wrong or they took an image wrong and they voluntarily said, you know what, my bad, I messed up. I'm going to do the right thing and take this. Some people do that, some people don't. But duplicate plagiarism, duplications. Like I said, Bruce said this graph before. So this is the number of retractions per year. This actually goes through 18. So this is pretty recent data. And I don't know about you, that's bad. That's incredibly disturbing to see that this. This is everywhere. This is not just the U.S. But the massive, number of retractions, and we'll talk about some of this in a minute. And then Bruce also talked a little bit about ORI, this Office of Research Integrity. Not to hate on anybody, but this is publicly available data. You can look this up online. These things come out, ever so often. You can see in 19, we've only had 3 of them. But in really, for lack of a better word, graphic detail. And I'll show you that this was one in this is June of this year. This comes out. This is the NIH listserv that we all subscribe to get. It comes out every Friday, and it tells us what grants are available and new policy changes, NIH and things like that. They've had this out for a long time. And so this is findings of scientific misconduct. And so what happens here is, they go in a lot of detail. I can't read it. Actually, I can read it from here. I got to take my glasses off. Let's see. Respondent knowingly, intentionally, recklessly falsified or fabricated Western blot data, blah, 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 blah. Falsely represented results using 64 different band images in 16 figures over all these papers. Now, what's interesting but also troubling at the same time is that this paper was published in 2011. Took till 2014 to get retracted, and this didn't come to light until this year. So you're looking at an 8-year window before this misconduct fully gets, out into the public domain, if you will. And so I don't know how to fix that. I don't know what we can do about stuff like that. But the fact that it's so, prevalent with those, publication numbers. Yeah, Brian.
So did you say that these announcements are part of the weekly digest? So if there's a finding of research misconduct, it's included?
Only, yeah. So that was, so this one was the one on January 28th.
Gotcha.
So, there's more down there. I just cut it off and everything. But yeah, these are actually, once they have the official finding, I don't know if they're required by law or whatever, but they publish it. So these are all discoverable, and they are, I don't know if you want to call it shaming, but it is what it is. They put it out there. So this is not anything you have to search for. You'll get this in an email if you subscribe to the NIH listserv. Like I said, it's only happened 3 times this year, but last year it happened 12 or whatever. But yeah, these are publicly disseminated, if you will. So this obviously had to do with image, manipulation. But obviously, retractions can come from many different ways. So this is actually, I'm not going to go into a lot of detail and everything, but this is a personal account. So this is some data, and every number that you see is red is made up. Every single one. Maybe not by a lot. Maybe this 1.67 was 9.67. Maybe it was 1.67 to the 5th instead of the 6th. But every one of those data points were changed to fit the hypothesis of what was going to happen in the experiment. And so it's not just simply, swapping a light band for a dark band in a Western blot, and everything, making the contrast different, stuff like that. It's things like this where you just tweak it just so much to where it makes it, where you want it to go. there's other aspects of it too. Data manipulation. So a publication has group A, group B, group C, and then person submits a grant. A is the same. B is now the data from C. C is now the data from B, but now C is not called C anymore, it's called D. I've seen this. This is a personal account. So this, this type of behavior obviously is not condoned by anybody, but it is prevalent. And it's frustrating for those of us who have had things happen to us, like the previous image and things like that, you try to run your lab with the highest level of integrity and transparency and reproducibility, and then you have people that are one-upping you by doing things like that. Incredibly frustrating. So all of these things, going back to here, outcry. We're fed up. Lots of opinion pieces. Everybody's mad. I found this one pretty interesting. This drug target slips away. This is actually something that I came across that I found was really, really interesting. Is that this is this ALS Therapy Development Institute, which I think is in Boston. And they took all these papers and they said, okay, the published studies showed that these are the percent changes. So this is how long the mice live based on all these different therapies. And this is what they reported. So this nonprofit place and everything did the exact same study with the drug, same conditions, and didn't reproduce a single thing. Nothing. And so when you look at drug targets slipping, it's because of reproducibility issues, right? Now, we know now there's all kind of things that all kind of things that can impact, right? Microbiome, the food, the bedding, whatever. There's a lot of things that contribute to this, but when you have, these outstanding results that can't be produced by a second party, it makes you nervous. I remember one of the in your career, you always have a bucket list. Two of mine have always been, I wanted somebody to come up to me at a meeting one day and say, hey, we did your paper for Journal Club. That was one of my, and the other thing is that I had one of my biggest competitors give a talk in which I was in the audience where they replicated my data in one of their papers. They took another, but talk about validation. I mean, that was fantastic to see somebody. And after, I went up there and I was like, hey, we're cool. We're not competitors anymore and everything. So it was, gratifying. But like I said, you see stuff like this and you think about, the lack of transparency, if you will, or the lack of, the rigor in reproducing these studies and things like that. And it kind of gives you some pause. So more blah, blah, blah. This is in, I think, 14, 15 and everything, Science and Nature, two of the biggest journals, out there saying, okay, we're going to start reinforcing, our methodologies and our, transparencies and everything. And I'll go into a little bit more detail about how that ultimately ended up happening. And so how does this happen? How do you not manipulate? I don't want to use that word, but how do you fool yourself into, thinking that, all this data is just totally legit? It's got to be right? And then, this was something that I'd seen before, 2015. And so you have this hypothesis myopia, collecting evidence to support a hypothesis, not looking for evidence against it. And so people often like to ask the questions that give us an answer. Yep, this is what's going on. Because it favors, what we want to see. So in my lab, we do infectious diseases in the lung. And, a lot of times, we do the proverbial toes up or toes down. They either live or they die. And if they die, then we fully expect their immune system to be messed up somehow. And so, so sometimes, that's what we see. And that's our yes. But sometimes we'll have where a mouse dies like crazy and their immune system is either not broken or over exacerbated. So again, it's not the yes, it's not the linear that we thought. It's more along the lines of, the curveball that some people, scientists in general, may ignore or may downplay. All right. And then the Texas sharpshooter, so seizing on random patterns of data, mistake them for interesting findings. I love the description of this. So the description is, so you see the bullet holes or whatever you want to call them up there and everything like that. So what they call this is that you shoot first and then you draw the bullseye around the closest one. I thought that's great. But it's funny how, we tend to do that. We're not infallible. Sometimes you'll see stuff all over the place and you're like, man, that's trending really where we want to do this. Let's do it again. We're getting closer. Let's do it again, and everything. And you always feel like, you're just chasing rather than, It's never going to happen. We're not going to get this to work. Joe is one of my grad students in the back there. He knows. We've done this in my lab many, many times where you're just like, it's if it's yes, no, or maybe, we drop it. But if it's yes, yes, no, 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 maybe, you find yourself chasing, and it gets really frustrating. But, like I said, you end up, what you try not to do is do the Texas structure. You try not to take the 2 yeses and the 1 maybe and ignore all the no's, right? You don't want to do that. All right, asymmetric attention. Checking unexpected results when giving expected ones a pass. And so this is also called disconfirmation bias. So when data doesn't match, we immediately think somebody messed up, right? So, in my lab, we have something called good hands, right? We believe your data if you have good hands in the sense that you can reproduce something, right? You can do an experiment twice and you get exactly the same answer. It doesn't have to be the exact same magnitude. This protein is made at this level and it's only 50%, in the second study, but the 2 studies look the same in the treated versus untreated, or the wild type of the knockout mouse, right? It's moving in the right, direction. So we look for reproducibility. So, this kind of goes back to what I said with the mouse that dies, and you expect the immune system to be messed up. And when it's not, maybe you tend to over-focus on what you were surprised by, rather than, thinking that it's not real. So, and then finally, this storytelling. Finding stories after the fact to rationalize whatever, and, everything is rational, right? I'm guilty of this. I'm not going to, lie. I mean, you get an answer, you reproduce it 3 or 4 times, it's the same answer, and then you're like, crap, I didn't expect that, or I really didn't want to see that. Okay, let's go to the literature, see if anybody else saw it. And we go to the literature, we're like, okay, let's see if there's some way we can rationalize while we're getting this, because it's consistent, right? It's consistent data. It's data that you've reproduced. It's data that you're transparent on. You wanted to see. And so you feel guilty about it, right? You feel like you want to go and justify it, and things like that. I think the take-home message is that you have to be comfortable in knowing that you did the right thing in the sense of doing the experiment, right? You're transparent, you're rigorous, things like that. So these are kind of, like I said, how we kind of fool ourselves a little bit in maybe missing some obvious, connotations in our data and the way we look at things that I'm not saying it all leads to falsification or all leads to misconduct, but, it kind of jades you a little bit. All right. So challenges to rigor, transparency, and reported science. Science is often viewed as self-correcting. And there's 100 articles out that says this, and there's 100 articles that say, oh, no, it's not. It's not self-correcting, So immune from reproducibility problems, pressure remains true over the long term. So I was trying to figure out exactly what this meant. I actually got this off the NIH website. So essentially what it is that hypotheses are abandoned when they are undermined by empirical evidence. So, you could have this one big paper come out, and it's celebrated, it's high impact journal, and everybody thinks this is awesome. And then a year later, 2 years later, 2 years later, and people are coming out and saying, not so much saying that it's wrong, but kind of filling out the rest of the equation and really finding out that it's not as simple as they made it out to be, or it's a lot more complicated, things like that. Ultimately, you get so far down the road and you're like, I don't think that's right? They're wrong, but it's out there, right? So we see that a lot. I probably a bunch of different fields here. I can name a lot of people in my field who has data that we just generally consider suspect. It is what it is. We just, and they're prolific, and they get a lot of publications, and they get a lot of funding, and you just kind of wonder, how is it? So short term and everything leads to, reproducibility problem, loss of time, whatever, and we'll go into that in a little bit more detail. So factors that short circuit this self-correction. So factors that basically block, this self-correction. So hyper-competitive environment, historically low funding rates, grant review, promotion defenses, high-profile papers. All right. So a couple things here. One, I'm a department chair, for 19 months now, and I don't care at all about high-profile papers. I really don't. I mean, I'd love to have a Nature paper tomorrow, but I'm not going to hold my data for 4 years trying to get it or manipulate my data. I don't expect my faculty to either. I mean, if you get one, I'm going to pat you on the back, but I believe in distributing information. I believe in publishing wherever, whenever, however that is. So I don't, we have to, the publisher perish, that's 100% true. You don't publish, you don't get grants, you don't get grants, you don't get more publications, you don't get tenure. It is what it is. That's the way our system is set up. And I believe in that system. But not to the point to where I want to fabricate data to get into that system. So historically low funding rate. So I just graphed this out. This is just data that I found online. So NIAID is the Infectious Disease and Allergy Institute at NIH. And you think about that graph earlier with the retractions going logarithmic almost, and you see that this is the pay lines, right? So 12% of the grants were funded in, what does that say, 9? And then 5 years later, it's down by 25%. So what if you had a faculty position here, right, and you're coming up for tenure here, and your chances of getting a grant have dropped by 25, 30, 40%. What kind of pressure do you think you feel?
Wow.
Right? The pressure to manipulate, the pressure to change 1.67 times 10 to the fifth, if 1.67 is 10, just say it, right? I might rationalize it. I'm sure as heck not justifying it. But, you can see where the pressure, comes from, and how rigor and transparency is anything but, right? Difficulty in publishing negative findings. It's funny because the lab I got my PhD in, we published a lot of negative data, and it was what it was. You did all the work, you hypothesized, you did everything right, and it was reproducible, and it was junk. It just didn't show much. And we published it, and I know I've had a revision on this opinion in my own career because, we have gotten to the point a decade ago or whatever where, we'd have a lot of negative data and we're just like, they're not going to take it. So let's just drop it. It could be years of research and tens of thousands of dollars invested, and you just flush it. You just don't do anything. And now I'm kind of getting back to where, no, it needs to be out. I got a good friend I went to grad school with. He's now he's the dean of research at TCU. And he posts a lot of negative data. And I'm like, dude, why do you always do that? And he's like, he said, for 2 reasons. One, it needs to be out there. People need to know that that this pathway does not play a role like that. And he said, and more importantly, I don't want anybody else to go through this. I don't want anybody else to have to do this, right? I wasted time and money, effort, blah, 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 like that. But I'm going to get something out of it. But I'm going to stop anybody else from going down this pathway and them wasting their time and losing their resources and things like that. It's a pretty good way to look at it, actually. So overemphasize, study big picture, finding, leaving out SAD sales experiments. We're going to talk about this in a little bit more detail in a minute. Like I said, big picture's cool. I'm all for that. But I don't want to. What's the term? Least publishable unit. I forget. Different people have different connotations. To what that means and everything. I look for stories. If I can find 5 figures, and we can do an A, B, C, D, E, and come up with a story, we're off to the presses. We're going. Sometimes you'll also get to a point where you're just like, what? I'll just have the reviewers tell me what I need to do next, because you get frustrated and you're like, this is beating my head against the wall. Where do I go with next? Things like that. So, Ron, you had a question?
Yeah. So the difficulty in publishing negative findings, is it difficult in that the journals don't accept them as much? Is that the difficulty?
I think that is part of it, but it's also, I think it kind of goes back to, the funding levels. They've rebounded, right? But there's more grants being submitted than ever before. I think productivity has become a bigger metric than it used to be. The other thing is, and I tell my faculty this all the time, is that, the stories and the data that I published a decade ago, I can't even get that to be reviewed now, much less accepted for publication. It's much more difficult. You have to do so much more in your papers now, much more mechanism, no descriptive, no corollary stuff. You've got to really, really, prove things multiple different ways. And I'll go into that a little bit in a minute, but prove things, ad nauseum, to get the reviewers to say, okay, yeah, this is a complete story. So I think, in that sense, negative findings, even if they are, or they appear to be in depth, at the end of the day, what are you left with? Negative findings. Nothing. So I think that kind of biases reviewers and journals against that. I'm not bashing. I'm just saying that that's like a human nature, if you will, in the reviewer world, right? So, and then finally, inadequate external design. This kind of goes to the rigor and transparency, inappropriate use of statistic. I actually just came across this word the other day. I think it's great. P-hacking. I love that. Everybody knows what a p-value is, right? So p-hacking is doing whatever test you can to give you the best p-value. I think it's funny, actually. I think it's I've never heard of it. I think it's an interesting concept. And every once in a while you'll get, I guess I'll call it illuminated. We got this, I don't know, 2 or 3 years ago where a reviewer came back and they're like, you're using the wrong test. Okay, sorry. What are you talking about? And they told us, and turns out that we're using the wrong test, but their test made everything all better. Their test made the data much more significant and look a lot better. And I'm like, golly, thank you. So that was reverse p-hack. That was the inverse of p-hack. That was hacking over the p instead. But like I said, in this sense, it's manipulative from a p-hacking standpoint, right? You're trying to find the best thing to help you sell your story and do your data. And then this is kind of the reported to resources and or unexpected variability. And I'm going to touch on this in just a minute and tell you a little bit more with that. All right. So finally, what are we talking about? What are these new guidelines, these new policies and procedures like that? So this came out in January of this year. Had to be incorporated in every grant, I think, starting, I can't imagine it would have been February 5, which is the first grant deadline of the year, because it came out on the 25th. So I'm guessing it was going to be for the June deadline is when this actually, where you had to start putting this stuff in. So there's 4 components to it: rigor of the prior research, rigor of the proposed research, biological variables, and authentication. So what are they? So what is rigor of the prior research? A careful assessment of the rigor of prior research that serves as the key support for proposed project will help applicants identify any weaknesses, blah, 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 describe the strengths and weaknesses, rigor of prior research, describe the plans, things like that. So what is rigor of prior research? The background. Why are you doing this? Why are you looking at fungal infections in the lung? Why do I do that? Why do I look at those? Tell me why it's important. Tell me where, the gaps are. Tell me what's not understood. Tell me what's dogma, and tell me why you think dogma's wrong, right? That's what rigor of prior research, that's what you have to address. And so I want to give you a concrete example of that. This is from a grant I have in review right now. It's getting reviewed in December. So we work on fungal pathogens. We work on asthma, because one of our organisms is associated with asthma. Chitin. Does anybody know what chitin is? Chitin is what's in the cell wall of crabs and shrimp and crawfish and insects. All fungi have chitin in their cell wall. We're looking at it from an allergic standpoint and everything. And so a couple of references here and everything. Despite these observations, a knowledge gap, keyword, buzzword, blah 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 blah. Collectively, prior research has left these things open, right? So we're basically, using the policy and using the buzzwords to our advantage. We're saying, look, this is what's out there. These are the holes that we think we found, and this is how we're going to plug it. So here's the holes, the rigor prior research, and then rigor of the proposed research. That's the hole that's being plugged. So you identify the hole, all right, knowledge gap, prior research, employed models that are tangentially. So you're setting it up why this still needs to be looked at. This still needs to be done a different way. That is what the NIH is asking reviewers and asking grant submitters to justify. It's not just about, these fungal infections cause, 100,000 cases a year and they die and everything like that, and the references are from 7 years ago. That irritates the heck out of me when somebody writes a grant and the references are old, right? I'll ding people on that, because you're just doing the same old thing. You're not adhering to the policies. You're not convincing me that this is a big deal based on data that's 7 or 8 years ago. So rigor, show me where the holes are. All right. So this is the scientific rigor, right? Application of the scientific method, robust, unbiased experimental design, blah blah blah. How the experimental design will achieve robust and unbiased. So this is In the old days, it was called the experimental design. Now it's called the approach. The research strategies, part of the approach, or the approach is part of the research strategy. All right. What's an example of this? So this is also from that grant. So what are we doing? So we're actually looking at this gene, this protein called AMK. So I'm not going to go into all the detail. But how are we looking at it, I think is most important. This is how you do rigor, in my opinion. This is one example of rigor. We have a mouse that is knocked out for this gene. We have a mouse that overexpresses this gene, and we have a mouse that can report where this gene is expressed. So I've got 3 separate models that I can interrogate the prior research, interrogate the gaps, interrogate the holes, the model systems in these 3 different mice. Other aspects are we're working on chitin, as I mentioned earlier. So in addition to just having a normal fungal strain that we infect our mice with or we expose our mice to and everything, we also have one that's mutated for chitin, has a reduction in chitin. So now we have 2 strains that are, target specific. And then we actually have purified the actual chitin from the cell wall of the fungus. And so again, rigor, how are you answering the question? How are you going to, greater depths and greater leaps to prove what you say you're trying to address? So that's where the rigor comes in. Here's another way we try to look at rigor. Rigor in design. So this could be in the design or in the preliminary data, however you want to do it. Verify, find, and use the complementary methods. Prove your hypothesis 2 different ways. This is the data we have. It's not published. So these, I'm not going to go into a lot of detail. This is a mouse that's deficient in this receptor for this cytokine. This is a mouse that's deficient in a receptor for a specific chemokine. This is an immune system protein that we measure, that we know in our model system is absolutely required for host defense. So if you don't have this protein, you die. The mice die. You don't die with the mice die. So these mice both have an increase in this protein. They both overproduce it, which was a little surprise. How can I prove this another way? What's an experiment I can do to reproduce this data in a different way? You didn't think I was going to test you. I didn't mean to do that because I used to hate it when I got that done to me. So how about we take the wild type mouse and we make it look like the knockout? So why don't we take the wild type and give it a soluble form of this receptor, which essentially blocks the receptor. So this mouse doesn't have the receptor. This mouse has been treated with a compound that blocks the receptor. Do we get the same phenotype? Yeah, we do. Same thing here. This mouse doesn't have the receptor. This mouse, which is the wild type here, was treated with an antagonist. Do we get the same phenotype? Yeah, somewhat. So we took our knockout data and we said, okay, let's be rigorous about this. Is this just something that's in the knockout? Is it intrinsic to the knockout? Are we only ever going to see it in the knockout? Can we prove to our reviewers, can we prove to ourselves that this is a biological one, this is a real finding? And so we took a mouse that has no issues with any of these genes or anything, and we made them have issues by treating them with these reagents. Again, rigor. It's all about proving, and transparency, telling people what you did. We're going to go into that in a little bit more detail here in a minute. Okay, biological variables. Again, I'm not going to go into this a lot because you have a lecture on this next week. Sex, age, weight, things of this nature. This is not something that we, traditionally deal with. And the reason why, this is actually a section from that grant where we talk about sex as a biological variable. 17 years now, we employ both male and female in experimental design. We do lung research. Animal experiments are not cheap. A normal wild type mouse from Jack's is what, $30, not including shipping. And we're going through $100,000 worth of mice a year in my lab. So, you don't buy these just for the heck of it, right? And so if you say you're only going to do research on one sex, you're just doubling your cost because when you could be using both sexes. So we've always used both sexes in my lab. Some people prefer males over females. Obviously in certain types of research, you can only use male or female, right? Prostate cancer, whatever, things like that. Cervical cancer, you can't. So, and that still is requiring justification, but it's obvious. Same thing, for, in this particular grant, we have some human subjects, and we talk about, that they're not biased or sexy. So you got to address this as, a new parameter. That is, scoreable. If you don't address these things, that can negatively affect your score. So you don't want to leave these out. And then finally, authentication of resources. This has been on, I think, about 2 years now. Chemicals, antibodies, biologics, all these different things. You've got to specify, where'd you get it? Catalog number. If it's an antibody, the clone, how much did you give? How often did you give it? Where did you give it? That is all required now. Not just the NIH applications, but publications, and I'm going to show you in just a minute. So this is from my grant again. 22 years before immunologic assays, you understand which commercial entities will provide the best products. That's true, right? I know some companies where I get antibodies from, and they stink. They're just not any good. But I know companies that have some really good ones. So we do not employ antibodies for blocking neutralization if they have not been specifically tested or documented by the vendor. So again, this goes with how do you authenticate? How do you know that when you block this pathway in the mice with this chemical inhibitor or this drug or this antibody, how do you know that you've done it, right? It has to be tested for that, and then you also have to show that you've proven that your pathway went down or something happened, some transcription factors stopped, whatever. You've got to go to different links to do that. And that's where the rigor and transparency, come from. All the mice are going to be genotyped. We're going to always measure to make sure they don't revert or they don't get mixed in with other strains or whatever. Just things like that. So these are things that are required based on this new thing with the reproducibility and rigor that the NIH has been putting out. All right. So how do we fix this? And I'm almost done here, actually. So how do we fix it? How do you stop from fooling yourself? How do you, try not to, bias your data, or how do you, try to be all you can be? Devil's advocate. Explicitly consider alternate hypotheses and then test them head to head. So you get an answer, you can come up with why it may or may not be true, and then you test both of those, all right? This protein is up, okay? It's up because it needs to be up, or it's up because it doesn't need to be up. All right, test them both. Figure out why. What's the impact? Pre-commitment. Publicly declare a data collection analysis plan before starting the study. So what that is transparency, right? That's it. That's the epitome of transparency. Be an open book. An example of this that's only come out since, I think, late 2016 is, anybody heard of STAR methods? STAR methods is now what all the cell family journals require. And other journals are starting to sign up to what it is. So you can see it here. STAR, structured, transparent, accessible reporting, right? These method sections of these papers are crazy. I mean, they are so detailed. Like I said, antibody clone numbers, catalog numbers, all the details. I find it so frustrating in my career over the last 25 years to go and try to use a reagent, and I find 10 papers, and I cannot find the timing or the dose or the route or where they bought it. We got these mice from Jack's. I can't find them at Jack's. I called Jack. We never had these mice. Where'd they get them? It's so frustrating as a researcher to not be able to track things down like that, And I think that's where this call to arms and this tidal wave, it started with fabrication or whatever. It started with retractions, and then it just, it's almost that was the gas. The fire was already a high level of frustration of just how hard it is to do this job and how hard it is to do science. Is having difficulty in repeating stuff because we don't have all the definitions. We don't have all the data. I remember one time we were trying to grow these cells out of bone marrow for mice, eosinophils, and we were using this protocol from this famous scientist at the NIH. We just couldn't get it to work, and we called him up and we're like, "I'm sorry, I don't know how you got this to work." We did like that, and it was 2 media changes on 2 different days that they didn't put in their methods. That's all we had to do. We didn't have to do anything different. We didn't have to isolate them differently. We didn't have to add different concentrations. Just change the media 2 more additional times. Worked like a charm. But they didn't put that in their paper. Now you've got to do that. STAR method says you've got to go massive ad nauseum detail, which I, for one, am happy about. Kind of a pain to do it as a writer because I just had to do one of these in a matter of speaking the other day for a paper that we just got published in JCI Insight. And they asked for, like I said, every catalog, every clone number, lots and lots of details. Statistical test. Every single time you used it, even if you were having to be redundant and repeating yourself, you had to put it where you used it. So we're starting to see this. The other thing that we're starting to see, and this kind of goes back to reporting your data and how reproducible it is. This is one of the graphs from that paper. Kind of hard. I probably should have made it bigger. But I want you to note a few things on this. This is a box and whisker plot. So you have the error bars here. Actually, they're not error bars. This is the box and whisker. The whiskers are, this is the max, no, I'm sorry. This is the 95th percentile and the 25th. The top line here is the highest value. The bottom line is the lowest value. The plus is the mean, and the line is the median. That's 5 different data points on one column. Because the journal required that we can only graph our data 2 ways. Column graphs, bar graphs, or verbatim. You could not use those. You either had to show all your dot plots, every single mouse, every single thing, or you had to do box and whiskers with all these components in it. Because when you do that, people know exactly how reproducible your data is, right? You have to be transparent. And journals are starting to do that. They're starting to say, where, look, you can't do just a bar graph with, a tiny error bar up there. You got to show a lot more detail. And so it was like when we were going through this review process and going through the process of putting together this paper, and you read the instructions to authors and you're like, oh my God, they're serious. you really have to do a lot more work now. But it's for the better, I think, and it's also because of what we're seeing. We're seeing massive amounts of retractions. We're seeing policy changes in NIH and everything. This is going to hopefully stop things like that, from happening. And so again, a lot of granularity in those images. I'll be honest. I mean, initially when we started doing it like this, I had trouble interpreting my own data when I graphed it like this because I'm like not used to looking at data like this. I'm not used to looking at these graphs. But once you do it enough, and you start seeing them, obviously you can see things that are, big differences, and things like that. So now it's like a welcome sight, if you will. We like to graph this way. So team of rivals, turn competition into collaboration, kind of what I alluded to earlier. I've got a story from 2 colleagues of mine. One's at Vanderbilt, one's at the University of Michigan. They work on the same pathogen. It's called RSV, respiratory syncytial virus. It's the bug that all the 2-year-olds get infected with every fall, and they get really sick and stuff like that. 2 competitors, friends, but very competitive people. One was using one strain, one was using the other strain. He'd get data, he'd get the opposite. He'd get data, he'd get the opposite. So they collaborated, and they see each other's strain, and now he started reproducing his data. He started reproducing his data because it was, a strain issue. And so they turned their competition, mutual admiration and respect, but still competition, into a partnership and saying, okay, look, I don't understand why you're getting that data. It makes no sense with what we're seeing, And so rather than, become an enemy and become frustrated, get together, figure it out, On that grant I told you about, we've got some data in that grant that is unexpected and really going the wrong direction in dogma. Not exactly the same models, actually not anywhere near the same model system, but chitin associated. And so the dogma is a cell paper. The cell paper or means paper? I think it's cell paper. And very respected lab, high level lab. And this first author is a junior faculty at WashU. And I reached out to him and I'm like, you've been doing this, for a while. We're into it now. I know our data is a little bit differently, but, I'd love to put you on my grant as a collaborator. Can we, and so we're doing experiments together. He's analyzing some data for us. We're kind of doing the same thing is that I don't want to, fight with this guy. I want to buddy up and try to figure out, what is our data telling us, versus, his. So, and then finally, this blind data, analyze data that looks real. That's a hard way to say it. This is the easiest way to say it right here. The less close you are to the data, the less likely you are to unconsciously hope for a specific finding, right? The classic example of this is trying to look at histology. Now you have a wild type and a knockout mouse, and exactly what the immunology looks like, or exactly the phenotype of the mice, and you want the histology to reproduce. And so you go and look at the images, and you're like, you're trying to search for the one that matches with everything else you have. right? So you're totally not biasing the data, but you're cherry picking, right? You know, and we've done this where, you know, you're like, okay, we've got these measurements, and the 2 images we're going to pick are going to be best representative of those measurements. You know, and we had a reviewer one time come back and said, no, I need something better than that. I need you to quantify your histology. And we did. Didn't match anything that we were showing. You know, we picked the 2 images that we thought best represented the phenotype based on all the other outcome measures. But once we actually quantified the entire lung, no difference. So we were embarrassed. You know, I mean, we're like, oh man, we kind of built this up. Now, don't get me wrong. There's a reason why you have Rigor. There's a reason why you do, you know, in asthma research. There's a reason why we do cell populations of the lung, inflammatory mediators, lung function, you know, all of these, you know, breaths per minute, you know, all these different things, because it's not just one thing that defines a disease, right? It's a lot of things that define a disease. So maybe when you do research, not all 5 things are going to toe the line. They're not all going to be, you know, down in your treated group. Sometimes 3 of the 5 are going to be down in your treated group. And that's biology, right? Not everything has to be down to have an effect. And so we ultimately decided to rather than show the image, actually we kept the images in as they were, but we were able to actually do some quantification, not of the images, but some molecular quantifications to support what we were saying in the images. And some matched, you know, pretty well. And so we ended up coming back and saying, you know, okay, look, instead of doing this non-quantitative or qualitative, you know, rendering of the pathology, we're going to do a quantitative version and measure these things that, you know, would predict what, you know, histology is. And so we ended up, you know, doing that. The other thing is that histology, one point in time, one time, right? You don't show histology from, you know, 15 mice over 3 experiments. You show one section of one lung or one liver or one heart at one time point in one animal. So, you know, it's a little tricky to really be able to convey what all that one image is supposed to show. So actually, that is all I have. And I told you we should be out here by 1 or 4, and I alluded to my height. But anyway, any questions? Any comments about any of the stuff I showed? Hopefully you got a little bit out of my grantsmanship. I hope it gets funded. It was only about 10 points out the last time, so hopefully we'll knock it in this time, but we'll see.
So, publishing negative results. Maybe I'm missing something, but it seems to me like if more people publish negative results, more funding would actually be available because fewer people would be chasing their tails.
I think there's some truthfulness to that. I think the more that you know, I think the more that it's published, I think at the least it would stop the goose chases, or it would stop, you know, it's almost like, you know, that graph where I showed where they had all the ALS drugs that worked at varying levels, and then somebody tried to reproduce it all, couldn't reproduce any of it. So it was almost, you know, what I take home from that is that all of these targets here are not the right targets, right? So we need to go and look at other things. So negative data would do that, right? You do 5 negative papers on some pathway or some model, and you're like, okay, we're missing. We are not barking up the right tree because all of this stuff is negative.
Or just so I'm in the fight, like, you know, months into the program, and as we are asked to come up with grant research proposals,
Yeah.
we don't know what's out there because we're new and naive. So it's like, you know, okay, what can I do?
Yeah.
But if somebody is already, or a lot of people have already tried this and it just didn't work.
You know, it's funny you mention that, because I have a colleague at the University of Colorado who sent me an email today, and he's like, look what just came across my desk. It was just the title of a paper that he got asked to review. And it is a project that I got funded as my first grant in 2003. And I never published it. And it was solid. The data was great. I was doing it when I was at the University of Pittsburgh. I moved to UAB. I could not get approved to use the equipment that I had at Pitt, at least not at the level that I wanted to at UAB. So I just kind of dropped it. So 16 years later, somebody has picked that up and has now written a paper on it, and I left money on the table, right? Because I never disseminated that information. I showed it a couple of posters at a couple of meetings and things like that, but I never, you know, and I got a grant funded on it, but I never got the data out. So had I published that paper in 2007, this paper wouldn't have been a realization now, and I might have had more funding on it. You know, I might have could have pursued it, you know, other ways. So, you know, I mean, you'll see this at meetings sometimes, especially the smaller meetings. where somebody will present data and, somebody get up and they're like, well, so-and-so, showed a couple of years ago that, you're wrong, this pathway's, invalid or something like that. And they're like, well, where do they publish that? I don't know, I just saw it at a meeting. Then that's not published, right? So now how are you to know that? You're not. That's hard to have that level of bandwidth to remember everything you see at every meeting you go to, right? But, I may have thought of the idea of doing this particular paper in 2003. I'm not going to get credit for it now. My fault. So, Ron.
Some of the real, true examples, instead of data that discovered were falsified, was it found because of the efforts in rigor and transparency? Is that I'm wondering if this helps prevent or research misconduct.
Prevention. That's a good way to look at it. Does this prevent research misconduct? I mean, in a way, if you're just going to make everything up day in and day out, then nothing's going to prevent that. You're just going to change it however you see fit. So no, it's not going to work. I think what it does is, things like star methods and things like graphing, the way we are encouraged to graph, not encouraged, we're told to graph now. I think things like that, will, stabilize, if you will, the scientific method in a way. I mean, we all know what the scientific method is, but, I think we're constantly redefining how to apply it and how to prove that we've applied it. So I think, prevention is a strong word. I'm not sure if we can, do that. I mean, like I said, everybody familiar with Retraction Watch?
Yeah.
Retractionwatch.com. I mean, it happens so often they even have like the gold star list. The guy on there, the number one guy on there is 250 publications retracted. So anyway, and that happens daily. So it's always going to be a problem. And like I said, when you're, and when you have, people whose jobs are based on whether they're funded or not, or promotion and tenure, whether you publish enough papers or got enough funding, whether you cover enough salary on grants, all those things, that's a lot of pressure. A lot of pressure. And so like I said, I'm not going to condone it. I'm not going to justify it. But, I understand the forces that push people in that direction. But you just, just got to be. I mean, everything we do is based on, truth. So I think, when it comes down to it, if you're, everything we do, everybody in this room is doing something for the first time that nobody has ever done. That's cool enough. Why lie about it? Why make it up? Why not do something, that people are going to, cite and reproduce and take to the next level? Like I always tell my kids, none of them give anything about science. Like, oh, Dad, you want to win the Nobel Prize? You want to cure? No, I don't do cancer research. Don't want to cure. No Nobel Prize and things. Well, what do you want to do? I want to publish. I want to get funded. I want to train and mentor, the next generation of scientists. And that's really it. That's what drives me. That's what I want to do. So it's not about the accolades. It's not about the, being on CNN or whatever like that. I don't have to do all that to justify why I want to do what I do. I get enough satisfaction just doing it. So, all right.
Thank you so much.
All right. Thank y'all.